Hi, I'm Rick. Some of you know me from other collaborations we may have done in the past, including GCCR. So I am really excited about this work I'm going to present today. All of this is published in, at least in the sense of it being out in preprints. And then there's a lot of future directions there that I'm excited to tell you about. My affiliation in the announcement is Arizona State and Google Research or Google Brain. And there's also something new here, which I won't be able to tell you much about, but I am and taking this research and putting it into a commercial entity and with some other olfaction people. And would love to tell you more about that later. We're still kind of in the stealth mode, so I can't tell you too much, but you'll hear more about it later, and I'm happy to sort of privately have one-on-one conversations about that if you're interested. Okay, so olfactory cartography. Cartography is the science of map making. And to me, this has always been like the most interesting part of sensory science is making maps of what the sensory world looks like. And so here you see in the lower left, it's a map of Middle Earth from Lord of the Rings, which I just rewatched with my kids over the last few weeks. I just love these like old maps like of that people would draw before they really knew exactly where things were. Right, you can see like in the upper edge here, there's some vague mountain range, and maybe it's not clear where the boundaries are. And that's how. And if you look at maps of for example, the New World, and were made in the 16th or the 17th century by Europeans, they have a similar appearance where things are distorted. There's blank spots. There's dragons where people don't know what happens. And this is sort of where I feel like we are with olfaction, is that our maps are extremely rough, and we like to make them sharper. And there's a lot of big questions that get answered about olfaction in the course of being able to make the map. So the map is not only, I think, valuable in itself, but I think the map is valuable as a test for whether we understand the thing we're studying. So by analogy to color vision, you have these very old kinds of maps of what color should look like, right? Like a color wheel. And you know what a color wheel looks like because you learn about a color wheel when you're 5 years old, right? It looks kind of like this, right? But this is like the pinnacle of at least the intersection of art and science as of 70 or 1700, is to have the idea that there are colors that are separate from each other, but the key idea being that colors that are perceptually nearby are nearby in the map, right? So red and pink here, red and orange. There's a perceptual nearness to them. That means they must be nearby spatially in the map. That's the key principle. And so this is state of the art at some time, and also what everyone learns today very early. Very, very simple, right? So how do people improve upon this? By 1930, there was an understanding of color as it relates to the stimulus itself, and also have maps that have some sort of perceptual calibration, right? So this is it's called CIE color space in 1931. And so what you see here is you have the same principle that colors that are nearby to each other, you would consider perception nearby if you are like a normal trichromatic person with no major variations in your cone receptors. And also you can see that wavelength is also mapped onto here. So the stimulus itself, right? You have 620 nanometers is red light, 540 nanometers is green light, and 480 nanometers is blue light, and so on. And you actually got a lot of interesting other things for free out of a map like this, which we'll go over in just a second. It is actually useful because you can do things like ask about how colors mix, right? Or how colors saturate. So for example, if you draw any line on a map like this, you can mix colors. So for example, if I draw a line between this color here, which is approximately pure 620 nanometer or 615 nanometer light, and this here, which is 465 nanometer light, if I want to mix colors, I can mix in proportions from this color to this wavelength to this wavelength, and I can generate any of these colors by mixing. Right? So it's very clear from this map exactly how mixing works. And in fact, you can now mix multiple colors. You can mix this color, this color, this color, this color, and this color on the triangle. And the interior points of the triangle correspond to all the colors you can mix from those three colors. And at some point, if you mix them at just the right balance, you get something that is fully saturated and is in fact white. So it also tells you about perceptual convergence, how you get to visual visual white from mixing a lot of colors, and among other things, right? And you can imagine modifying this diagram to reflect people who have variations in their color vision. For example, if they have a variation in the X-encoded clone receptor, or the clone receptor on the X chromosome that men often, is why so many men are red green colorblind, or even tetrachromatic women who have multiple functional copies of that receptor and have essentially 4 channels for color vision. So there's a correspondence here between the stimulus and the percept, which is really important. You can take a stimulus, pure wavelength of light, a photon of a given wavelength in the real world, you can map it onto the percept as you experience it. You get rules for mixing, and this is extremely tractable, right? You don't even really need a model. You almost just need a ruler to figure out what's going on here. Now, it's not perfectly, it's derived from psychophysics, right? So no one had to go out and measure all the properties of receptors. This is 1931. No one even really knew that there was such a thing as a cone receptor, but they could work this out just by doing experiments on color matching with human subjects. There are modifications you might make to this, right? So you might have noticed that in this first example on the left, you have some areas where there's a perceptual invariance of very large regions and very small regions. So up in the top here, there's many, many colors which to a normal person look approximately green, right, with not a whole lot of variation, and other parts of this map where the color changes very rapidly as you move a very small distance, right? And so in modification to this space, they give you perceptual invariance and sort of stretch out the space so that any given chunk or size of this map is equal to colors that look approximately the same, or there's a certain discriminability radius that is constant over the map. So in this modification in the 1976 color space, you get more perceptual invariance here, where each circle here has approximately similar differences in color across its extent. Okay, so what does this have to do with smell? So I would like to do something like this, right? I would like to be able to make a map of smell. I'd be able to map a molecule into that map. I'd like to be able to ask what that molecule smells like, and I'd like to be able to ask how those molecules can mix together to make other smells. So here's what I call cargo cult science, right? So cargo cult means people who are trying to approximate the real thing by emulating its forms, but not actually getting to the function, right? And so this is the form of a smell wheel that, it captures. It's not that it captures nothing. It's not that you wouldn't put, onion, garlic, and cabbage together in smell, but this is really just trying to fool you into thinking that we understand the mapping of odor without actually having any true understanding here. So I want to reject this as an option. So we actually need to learn the map from data, probably from a lot of data. And the first step in that is to turn molecules into rectangles. So what do I mean by that? I mean that in machine learning and basically almost any mathematical operation that you might do to solve this problem, you have to take some sort of input that looks kind of like a matrix or some sort of two-dimensional array. There's sort of like an input to a lot of kinds of operations that you do in math and in ML. And so the first step is, how do you take a molecule, which is not like, it's not like a spectrum, right, that you might have from looking at the color, we have a spectrum of light, and you have the intensity at each wavelength, right? That would already be just like a nice clean vector to use. This is a molecule. How do you turn a molecule into something like with a fixed representation? So this is, sorry, this is more or less what I said. We want to turn molecules to look something like this. It has to be like some sort of rectangular shape that has numbers in it, that is a representation, some sort of invariant representation of that molecule that we can use for learning. So the hard part is that molecules can be all different kinds of sizes. They can have all different kinds of rules. They might have sort of uncertainty about certain information about bonds. There's different atoms at different nodes, different ways, different kinds of bonds, single bonds and double bonds, and of course, you see all different sizes. So the trick, and this trick is shown in a preprint from Google Research from 2019, and I think I might have the date wrong at the bottom here when I show it, but the basic idea is to use what's called a graph neural network, and the graph neural network does the following. It takes a molecule, like say vanillin, and it turns that into a graph representation. So the graph representation turns atoms into nodes, and it turns bonds into edges. So essentially, it's just a representation of who is connected to who and how. Okay, so every node can be one of a handful of atoms, carbon, hydrogen, oxygen, sulfur, nitrogen, and so on. You don't have to go to the entire periodic table. There's no odorous molecules made of, gold or rutherfordium or anything like that. But if you, so once you've constructed that graph, you do what's called a message passing algorithm. So the message passing basically says, okay, let's have an atom right here, and let's say it's a carbon atom. What we know about this carbon atom is how it's connected. It's connected to the rest of the graph by having an edge here, which is a bond, an edge here, and an edge here. What if we share this information with the atoms that it's connected to? So we're going to sort of diffuse or distribute information about connectivity from this node to its neighboring nodes. And that would be like one step. And the next step, we're going to diffuse it to another set of nodes. So the information, the local information within the molecule about how it's connected is going to basically diffuse in a series of steps to its neighbors in the bond structure of the molecule. And at the end, essentially, you just add up all this information. And what it gets for you is you get a fixed length representation of a molecule. So no matter how large this molecule was, you're going to get a fixed length, and that fixed length is something you get to determine as the person designing the model. It could be length, in this case, it's showing length 10. It could also be length, at 64, it could be length 250 or whatever you want. The bigger you make that final representation, the more information you can encode about the molecule. But if it's too big, you tend to overfit, and you need like an extra large amount of training data to learn it. What we found is that numbers somewhere between 64 and 256 were a good size for this representation. And then on this representation, we attach what's called a prediction head. So this is just a way of encoding the molecule in a fixed length vector, right? So no matter the size of the molecule, you always get the same size vector. And then we have a prediction head at the end, which is to perform a specific task. So the specific task this model is initially trained to do was to predict odor labels from the Leffingwell, the Leffingwell data set, and from Good Sense. Good Sense is a place you can visit right now on your computer. It's a website that has odor labels for about 6,000 or 7,000 different molecules. Leffingwell is a similar size data set. Altogether, when you remove the overlap, you have 8,000 or 9,000 molecules to train on. And you can ask, can this kind of representation of a molecule be used to predict these descriptors? Can you predict, is this molecule citrus or spicy or beefy or so on? Okay. So that was what was done in 2019, and I had only a small part in this. My part in this project was to create a baseline algorithm, not based on this, for this model to beat, and did so handily. So essentially my role in this was to be a person who has some idea of what they're doing in making a predictive model, and then for them to show that their approach is better, which it was.
Rick?
Yes.
You ask us in case we have questions, and
yes.
We have one question on the chat. So
yes, please answer. Please
answer. Maya would like to know if you can take into account stereoisomer, so chiral center in the molecule.
Yeah. So if it's an enantiomer, the answer generally is no. Enantiomers, I actually consider enantiomers. If you want to talk to me about this later, I would love to talk about it. I consider enantiomers for most approaches to be like one of the most interesting things we can study, because most approaches, including this one, will generally fail to distinguish between two enantiomers, even though we know many pairs of enantiomers have a divergent odor character, right? So that's a super interesting question. But no, this generally, and actually I can't say with 100% certainty, but I'm almost sure that if you give L-carbone and D-carbone, and you put them into this model, you'll get the same output representation. Does that answer your question?
I'm not sure she can ask you directly questions, so that's why.
Oh, okay. Yeah. So yeah,
she said thank you. Think it's okay.
That's a blind spot. Chirality is a blind spot of this approach, and other approaches that I've seen, including using physical chemical descriptors like from Dragon or Morgan fingerprints, Dragon, Mordred. All of those will give you the same number for like D-carbone and L-carbone. So you cannot actually distinguish them from many approaches in ML that try to make odor predictions, which is one way we know that this approach is not fully complete.
We agree.
Okay.
Okay, thank you.
Yep. So why is it hard to do this? The problem is like this problem in general, right? Why does it just not work? So one reason is was given by that question in the chat, but another maybe bigger reason is that structure is a poor match for odor. Just what it looks like structural relationships that should encode odor are not in fact so accurate. So and again, some of these slides you may have seen before. If for example Joel Mainland or Alex Loshko has given a presentation publicly, you may have seen some of these before. So I apologize if you've seen them. We like to share a lot of the best slides. So for example, here's lyrol. Lyrol is a molecule that's used in perfume. Lyrol is essentially not allowed to be used anymore by perfumers. I mean, the interesting thing about perfume, there's not like a regulatory body that says you can and cannot do this. They're essentially self-regulated, and they make decisions about molecules that they are going to choose not to use anymore. Lyrol is one of them, and lyrol is not used anymore because of concerns about toxicity. So you might imagine if we have lyrol and it has this odor character, muguet, fresh, floral, and sweet. How would you make a molecule that's, what other molecule could be used that would smell like lyrol? So you can imagine some molecules that might get kind of close, right? You could have something where you just remove one carbon and it has some of the same properties. That would be the first entry here in B. You might imagine a molecule that is just a little bit more complex, and you've changed this aldehyde here to this like lactone structure. You get muguet, fresh, floral, sweet. Okay, that's kind of close. Okay, great. Okay, but what about this right here? You just move this bond here over to here. It almost looks the same. If you don't really look closely, you almost think this molecule is the same as this molecule. But this molecule is odorless, right? So that's quite a big difference, right? From having this be a great molecule for perfume. You can also radically change the structure and get something like this that looks almost nothing like the original structure, and yet has almost the same odor character. Right. So you have molecules that have both perceptual cliffs and structural cliffs. So the term activity cliff is used in pharma to mean a change in a small change in structure that leads to a large change in activity. Right. So someone might have a drug and they want to make a new drug that's similar to the old one, and they make a small modification. All of a sudden, the binding affinity goes up by 4 orders of magnitude or goes down by 4 orders of magnitude. So it's called an activity cliff, and people in pharma are really interested in trying to predict when these things will happen. And here again, you have here, this is an odor cliff where the odor changes by a huge amount despite a small change in structure. And this is the inverse of that is a structural cliff where with a small change in odor despite a very large change in structure. So because of problems like that, and there's many examples I could show that are like that, it makes it hard for these algorithms to work. And one reason why computing simple fingerprints like just taking generic like little pieces of substructures and using those as as elements of a predictive model, or computing physical chemical properties that you can get from normal off-the-shelf computational chemistry software. One of the reasons those don't work is that molecules like this and molecules like this are often too similar to each other in those ways. And these message passing approaches of graph neural networks do a better job. And it's hard to explain exactly why, but they do a better job empirically of distinguishing them.
Rick?
Yes.
There's one question. Tanoi asks, how do you think about utilizing chemical mixtures, a chemical mixture data set with multiple odor descriptors to create the odor map? Is this something you want to answer right now?
I'll get to that at the end. All the things I'm going to show you here are single molecule work. Mixtures is very clearly the frontier and the place that everyone is going to be going, because we're going, but the models I'm presenting here are by no means perfect, and there's always room for improvement. But being able to answer questions about mixtures is super important. There's only a few like big academic papers that show that, like from Sobel Lab, for example. But the reason mixture data is harder is essentially to really crack the problem to the level that we've done for single molecules requires at least 10x the data, right? For complex mixtures, maybe quite a bit more, right? And so even the models we see in academic papers that have say 50,000 or 100,000 data points are inadequate to truly solving the problem. And so it just essentially will take a lot more data, right? Which is actually one reason I'm moving on to a commercial application where we can actually collect that kind of data. So sorry, I won't be talking about anything more about mixtures today, though.
Okay, thanks.
Okay. So what can we learn about structural motifs and how they drive it? Can we learn like there's a piece of a molecule that tells you something is vanilla or tells you something is chocolate, right? So basic approach here, right, as I went in the previous slide, is to take each of these molecules and turn them into graphs, turn those graphs into fixed length vectors, and to add a prediction layer on top and to produce predictions about odor descriptors. And what that does for you, because you get a fixed length vector, that fixed length vector or array is just really a pointer into space, right? It's a location in a space. You can take that space and you can reduce the dimensionality of that space to make a map, right? You could look at. You don't have to reduce the dimensionality. You can deal with 120 dimensional space or 200 dimensional space if you want for some set of applications. For other applications, you might just want to actually make a reduction, make an embedding into a lower dimensional space. That's what's being shown here. So you take a vector of a certain length, and we can map it onto a particular point in, say, 2-dimensional space or 3-dimensional space. It will lose some of the information naturally because it reduces the dimension, but it might nonetheless be a useful way to look at the map. So, so that's the first step towards mapping. So I just want to present how did that approach do? That approach compared to a baseline showed improved performance for essentially every odor category. That improved performance was only loosely related to the amount of training data that we had. So there is a certain number of molecules that have the label terpenic or have the label fruity, right? So lots of things, especially in a data set you get that most data people collect has been focused on flavor and fragrance because it has commercial utility. And so you get a lot of fruity molecules, you get a lot of green molecules, you get fewer terpenic molecules, right? That's not as much in demand for what people want to put on their skin or in their food. So you have many more examples of these things like fruity. Nonetheless, even just for varying numbers of examples, you seem to generally solve the problem significantly better. AURC, for those who aren't familiar, 1 means you're doing perfect prediction. You perfectly predict every terpenic molecule. You never say a non-terpenic molecule is terpenic. You never say a terpenic molecule is non-terpenic. That would be AUC 1. That's pretty good. This is less good, right? So if down to sweet at 0.75, that essentially means that if I took a random pair of molecules, one sweet and one non-sweet, I'd have about a 75% chance of telling which was the sweet one, which was not, which is certainly better than chance. Chance is down here, 50%, right? You never do that badly. But it's also, leaves a lot of room for improvement. Okay. So the problem and the reason why all these, I think the reason why all these are not much further up on this curve, right? So this is the model we tried. This is described in 2019. This is the baseline I had made. It's based on the DREAM olfaction challenge paper that's published in 2017. The reason these numbers are not all up here, I think, is because there's still not enough training data. So you might say, well, hey, you had thousands of examples of molecules. How is that not enough training data? It's a lot of data. That's really not a lot of data, right? So problems that have been solved in machine learning have much more data than that. So take vision, for example. Take computer vision. ImageNet, which is by no means anymore the frontier of visual of an image library, has about 14 million hand-annotated images. Which that means is that people took images that were, say, they were fixed size, like 256 by 256 pixels, and people put labels on them. Let's say, okay, this is a bird, this is the sky, this is a stop sign, and then people use those to classify those images. So they get a new image. What does the image contain? Does it have a stop sign? Does it have a bird? What does it have in it? That's a pretty large library, 14 million, right? That's probably at least 100 times larger. It's maybe, or actually it's more than that. It's 1,000 times larger, probably at least, than anything we have in olfaction for single molecule data. It's 1,000 times a huge amount. And the thing about size is it's everything, right? So the thing about modern ML is it's really greedy with data. So sometimes if you have a model like, let's say, do a traditional machine learning, like it's a support vector machines or it's a linear discriminant analysis or something like that, it might work well with a certain amount of data, but then performance plateaus. And things like neural networks tend to actually be worse when you have very little data, like hundreds to maybe thousands of examples, but eventually they take off and eventually become much better. And this is maybe exaggerating the difference in the performance, but the deepest neural networks that have the most parameters don't do well until you have, millions of data points. This is also shown again here. There's the scaling where there's a number of examples that you have increases. The accuracy, which is shown in the y-axis, is essentially logarithmic in the number of examples that you have. And so I had one more slide here, which is not shown, but essentially, if you look at all of the major breakthroughs in ML over the last 10 or 15 years, in almost all of them, the big breakthrough was getting a really large data set. So if you look at superhuman performance in game shows like Jeopardy, or you look at Google Translate, or you look at AlphaZero, or whatever it is, right? The innovation was the ability to get millions of examples of the task and to put those into the model. It was not like a tweak to the algorithm. The tweak to the algorithm was somewhat like less important, or at least was something that was developed 10 to 20 years ago, but couldn't be used until we had A, a ton of data, and B, a ton of processing power. And for the problem like olfaction, actually the limitation is not processing power. A lot of these models you could run on a desktop with one CPU, but it really is the limit. The limiting factor is the amount of data you have. So my proposal is that pretty much no matter what you do, if you're anywhere near the frontier, if you're using any reasonable model, if you had 10 million labeled data points for molecules and what they smell like, you're going to beat everyone. Anyone else who has 10,000, no matter what model they're using. So getting a lot of data is really important. So how do we get a lot of data? The funnel actually for what you can do gets narrowed down somewhat quickly, right? So this is from there's vendors from which you can look at very large chemical catalogs. This is like an underestimate of the number of distinct molecules you can order. It's this is like one catalog that has about 7 million, but their website, their sites that you can do custom synthesis for you, and they have, like 100 million or more molecules. You can filter that down by certain simple rules about what's likely to have an odor, and then you can filter down for things that are cheap, right? So if you don't want to break the bank in your experiment, you don't want to order molecules that cost, $100 a gram or something, or $1,000. $100 a gram is fine. $1,000 a gram, okay, now it's you're starting to reach your limits of your budget. They also have to be non-toxic, so you have to filter out things that have known problems, right? And then, okay, so from here, you could probably order almost any of these things with some delay. In the case of the data I'm about to show you, and here's this test real quick that's slightly misleading. 400 is just what we chose. There's a larger catalog we could have chosen from. We chose 400 novel molecules to test this model. So what is it that we're doing here? Most literature that you've seen does retrospective validation of a data set. They go to, for example, Good Sense, or let me skip ahead here. They go to, for example, Good Sense or Leffingwell, or some of the data, Dravnik's Atlas of Olfaction. Some data set, they take that data set, they build a model, and they fit the model, and maybe they do cross-validation to make sure they're not overfitting, but still, they're fitting within distribution. They're fitting to the same kind of data that they built the model against. That's normal, but we decided we would do a prospective validation test. We would collect entirely new data. We'd freeze the model in place the way it was in 2019, and we test on this entirely new prospective validation data set to see how well we were doing. This is a real reality check, because a lot of models that do well, even in cross-validation with the data set that they've been trained on, suddenly perform poorly out of sample or out of distribution when you bring new data to bear on the problem. Right? This happens all the time. So we want to see if this would happen for us. I'm going to skip ahead here. Sorry, I'm not skipping ahead here. Is this map for olfaction? Okay, so I have this embedding space here. This embedding space here is a place where you can place any molecule after passing it through the graph neural network transformer that transforms the molecule's representation to a fixed length vector. Can this be a general map for olfaction, like a map that can work for all olfactory problems, right? That's a pretty bold claim, because to be a general map for olfaction, it would have to mean that there's like some single representation of molecules in olfaction, not only for all kinds of olfactory tasks that a human might have to perform, but possibly for all olfactory tests that any animal would have to perform. So it seems a little bit unreasonable. So how general can it be? One test of this generality is to do transfer learning across different species. So humans and mice separated evolutionarily like 100 million years ago. So you can ask, if we train a model and build a map that is entirely based on human labels given to odorants, and then we ask mice, what do these odorants smell like? How well will we do? Will things that are positioned close together in the map smell similarly to a mouse? Will they activate similar representations in a mouse brain or not, right? And if we go further back in time, 500 million years ago, evolution diverges to insects, which have an olfactory system that has evolved independently, uses a different kind of receptor, uses ionotropic receptors instead of G protein coupled receptors, and have a kind of a similar kind of olfactory structure, but that has evolved completely independently. And what will we see? Well, insects respond similarly to odorants that are positioned in a similar way in the olfactory map. So there's a lot of reasons why this might not work, right? You have different kinds of olfactory receptors. Oftentimes in chemistry, generalizability into different chemical classes is weak, and to different kinds of tasks, right? If you ask whether two things have the same boiling point, doesn't necessarily predict whether they'll have the same pKa, for example. So that's reasons why it might not work. Reasons why it might work are that because of convergent evolution, all of us and all of our animals and all of our insects evolved in somewhat correlated, especially terrestrial animals, odor environments, right? So you and a mosquito, you have different olfactory systems, but you still live on planet Earth. There's still olfactory tasks, and there may only be so many ways to solve this. And of course, the way that the olfactory system is implemented doesn't necessarily determine what the computation is that's being done. So what we did first is we, by the way, how am I on time? Should you give me a time check?
You have 25 minutes left, including questions.
Great, I'll take about 15. Thanks.
Right.
What we did is we took a reference data set. The reference data set is the data I was just talking about, from left and well and from good sense, about 7,000 molecules. We train this graph neural network to get a fixed representation, a fixed length representation, which we're going to call the principal odor map. So I'll refer to the principal odor map a few more times here in this talk. And then we ask, how does it perform on other data sets in human olfaction, in animal olfaction, and then in non-olfactory tasks? We ask what's the performance. So what does performance mean? Performance means we start with these molecules and we get a fixed representation, a fixed vector for every molecule. And that fixed vector is determined by training on the human data. If we take this fixed vector and just attach a new prediction head to it. So for example, we take the fixed vector and we perform a simple algorithm, not a deep neural network, but a simple algorithm like random forest or support vector machines onto this representation. How well can we predict these tasks? Okay. So they're essentially asking if we have a good starting representation. If we have a good starting representation here, that means that essentially we could take any molecule, put it through this pipeline, and get a fixed representation of that molecule that's useful broadly for all olfactory prediction tasks, right? It's a big challenge. Okay. So what this looks like is we start with the human odor data set. We pipe it through this model, get the map, and the simple model, like say random forest, gives you prediction, right? That would be the pre-training where we actually are modifying this model. We freeze this model here, and now that it's fixed, we simply pipe molecules through this model, frozen, get a frozen representation of that molecule, and ask whether a simple model can give us an accurate prediction. So it's called transfer learning. Okay. So we also compared a few baselines to see if what we're doing is going to do better at this task than alternative approaches, right? So two popular alternative approaches. One is to compute fixed fingerprints of models. Fingerprints essentially means you break the molecule into little fragments. You ask what those fragments look like, and if a molecule has that fragment, it gets a 1. If it doesn't have the fragment, it gets a 0. And you string together all the 1s and 0s for all the possible fragments. That's what molecules as fingerprints means. It's something called Morgan fingerprints, and it's a standard approach in a lot of software and in a lot of other chemistry tasks. Also, you can compute physical chemical descriptors. You can look at the molecule as a whole, and you can ask questions about that molecule that are computed by computational chemistry software. The kinds of features or descriptors that you get are sometimes very interpretable, like the things like what is the predicted boiling point of the molecule, what is the log P of the molecule, which is a measure of lipophilicity. Sometimes they're really weird things that no one knows what they are, including most chemists, but nonetheless, they are often used in prediction tasks for getting fixed length descriptions of molecules. So we compared against both of these to see if our approach could do a better job of transfer learning to these. Now note that these approaches here, they're not specific to olfaction. They're general for any sort of chemoinformatic problem. Whereas our model is trained on olfactory data from humans. So one reason we may expect our models to beat the baseline is that if that olfaction in general is a sort of a class of tasks that if you train on one task, you do better on other tasks in olfaction, even including across species. Whereas if you do these sort of chemoinformatic representations, you're not like capturing the essence of olfaction, but you would do worse. So what we found was that our model, the principal odor map, which comes from the graph neural network, was better than the sort of generic features I described in the last slide for all olfactory tasks, but not, or almost all olfactory tasks, but not for chemoinformatic tasks. So, for example, we asked how well we predict the Drosophila behavior, how well we predict or binding in mosquitoes, how well we predict in other human data sets for behavior, how well we predict neuron activation in Drosophila, how well we predict neuron activation in mice, and so on. And for all of these tasks across multiple species and phyla, we perform better almost always when we have when we use this principal odor map than when we use these generic chemoinformatic features. But there's a trade-off. You actually perform worse at chemistry tasks. So there's sort of like a no free lunch theorem in ML, which says, you know, if you have a representation that allows you to improve performance on one set of tasks, you probably get a degradation of performance in other set of tasks which it's not tuned. So if you want to predict things like overall binding affinity to other kinds of receptors, or to receptors that are not in that are maybe related to in the same class as olfactory receptors, but they're not olfactory receptors. So there are other kinds of GPCRs besides olfactory receptors that are found in the body. We do actually worse in those tasks when we use the principal odor map. So it's better in that case to use a sort of generic chemoinformatic feature. So this odor map generalizes across 500 million years of evolution because it's the best way now to take a molecule, run it through this pipe, and now ask, like, how will a mosquito respond to it? How will mosquito receptor? How will mosquito behavior? How will Drosophila behavior? How will Drosophila neurons, right? Human neurons, mouse neurons, right? Is better than alternative approaches that have not been trained on human perceptual data. Okay, so the map itself has some interesting properties, right? So you can look and you can get sort of a hierarchy of representations here. So each dot is one molecule, and here we're showing the embedding, a two-dimensional embedding. So you take this fixed length vector that we are calling the principal odor map, and also you just say the graph neural network, and we reduce this to 2 dimensions, and we place every molecule that we have in this space, and then we draw the contours of molecules that had as a label things like muguet or lavender or jasmine, which are all floral notes, or savory, beefy, or roasted, which are all meaty notes, and so on. And you notice that there's a hierarchical structure here. So it's not only the case that there are stereotyped places where, say, lavender or jasmine live, but that they're nearby to each other, and they're part of a sort of supercluster that's floral. Whereas the meaty clusters are part of a supercluster that is the meaty subclusters are part of a supercluster of meatiness, right? So you recapitulate this hierarchical representation, and you don't get that using chemoinformatic features so well. And what we noticed is that this is quite similar to if you look at things in these super categories, they actually represent different things that happen in the real world, right? So things that smell fruity are a product of a fruiting process that happens in nature. It's a metabolic process of fruiting that comes to starch and photosynthesis. Floral things are a product of floral metabolic processes, and some of these nastier things are a process of putrefaction, combustion, fermentation. And then, of course, animal respiration is here as well, right? Animals breathing, that's a metabolic process that can produce, or I'd say not breathing, just animal respiration in general can produce things like the musky things you get from like musk sacs in an animal. So there's this interesting hypothesis that the reason things have this structure is that they're actually related by shared metabolic processes. So the hypothesis specifically is that if things follow shared metabolic pathways, then they should smell similarly. If they have very different metabolic pathways, they're very metabolically distant from one another, then they should smell differently. So to be more specific, we took a database called Metapsych. Metapsych is, some of you maybe have heard of KEGG before. Metapsych is like KEGG, but it's a little bit smaller, but it has higher quality data. So almost all of the data from Metapsych is elucidated from experiments. So they take there are experiments that ask essentially whether a certain molecule is converted into another molecule as part of some metabolic process, right? And hence it labels these metabolic processes. Where do they come from? Do they happen in a flower? Do they happen in an animal? Do they happen in a human? And from this, you can actually construct a metabolic graph, right? So this molecule might turn to this molecule, might turn to this molecule, might turn to this molecule over the course of some chain of steps in some metabolic process in some organisms. So you can construct this graph, and you can ask, what is the metabolic distance within this graph? Again, we haven't said anything about odorants yet. This is like all kinds of molecules. But if we take 2 odorants, so here I'm going to label the odorants in color and some non-odorants in gray. So if we want to go from one odorant to another odorant, sometimes you have to go through a chain of reaction steps that includes non-odorants. Nonetheless, that in this case, going from the yellow molecule to the blue molecule is 3 steps. So here's the metabolic distance is 3. So the hypothesis then is that the metabolic distance, how metabolically related two molecules are in the real world, is going to be related to their olfactory similarity. And specifically, we predict that the metabolic distance is low, meaning if there's just a small number of steps to link two molecules in some animal or plants or other organisms, a metabolic network, that things will smell similarly. And if there's a large number of steps, they'll smell very different. And because we can't go out and measure the similarities and differences of all the tens of thousands of possible pairs of molecules that are there, we're going to use distance in the primary principal olfactory map as our measure. We know that distance in the principal olfactory map is already related to odor similarity, right? Things that are lavender are close together. Things that are lavender versus things that are savory, those things are far apart, right? So we're going to use that distance as a measure of similarity. And what we get here is this remarkable correlation between metabolic distance and distance in the principal odor map. So things that take many, many steps to link them have very high distances. Things that take very few steps have very low distances, and the correlation is quite high. For alternative representations, you also see a relationship, right? It should make sense that if you go one metabolic step, you can only do so many things to that molecule. You can only remove so many functional groups, right? So you should imagine no matter what model you chose, you might get some sort of similarity in odor, but the correlation is much weaker and noisier. So just to make this concrete example, you could take 2-pentenyl and 2-pentenol, which by most feature calculators, for example, using morphing fingerprints, using physical chemical descriptors, will be considered to be very different because you take one of the major functional groups and you change it. In this case, from an aldehyde to a ketone, to an alcohol. Primary alcohol, which seems like two very different things, but in fact, there's actually a very similar odor character, and they have very similar representation in the principal odor map, and they are linked by one metabolic step. Here's another example between thiamine and sulfurol. Most ways you can compute the difference between these molecules would say these look very, very different. Why? Because you've gone from thiamine, you've lost half the molecule, right? This giant chunk here, and maintained only this backbone. The principal odor map says, no, these are still very similar, right, to each other, which has been trained on human olfactory perception, and is claiming that these molecules should smell similar to each other. In fact, they do. They smell meaty, roasted, and cooked. They're also linked by one metabolic step, but they have a very large distance a large edit distance in these conventional ways of comparing molecular similarity. You can go through entire reaction chains, right, and for example, these biosynthesis pathways. And if you use the primary odor map, you actually trace out fairly smooth paths through the space, right? So it's as if the reaction is going from somewhere to somewhere in sort of a directed fashion, right? From the things that smell like this to things that smell like this, right? If you do this in a generic chemical space, it sort of bounces around, right? It doesn't have a clear direction, right? So it's as if as you go through metabolic space, you're going through the olfactory space concurrently, right? They're very closely linked to one another. That's the big claim of this paper. I'm going to skip that. It's just more about the same thing. So the principal odor map is strongly correlated with nature's metabolome, and so our claim is that the origin, the evidence of what I showed you there, the larger speculation is that the reason any two things smell similarly. And if you give an implementation reason like, oh, they share a common receptor or they activate the same receptors, but why do they activate the same receptors, right? We could have had any receptors at all. Any organism could evolve just about any possible receptor over millions of years of evolution. Why do they evolve the receptors that they do? And why do those receptors lead to activation that gets correlated in a certain way in the olfactory bulb, in the peripheral cortex, or in the antenna lobe, and in the mushroom body? Like why do those correlations exist over evolutionary time and over developmental time? And so the argument here is that it's all related to trying to be a metabolic state detector, right? That if you smell something similar, that two things that smell similar, they smell similar because you've evolved to a metabolic state detector, and things that smell similarly are in some related metabolic state. Okay, I'm going to skip this part and move to this. Okay. So this principal odor map is tuned by odor statistics in nature. And you can see here, what the figure on the left is basically the figure I showed you before. I just rotated it. It just got rotated like 90 or 180 degrees. If you try to do this with other representations, you get things that are very harder. You don't recapitulate the same hierarchical structure. So if you use Morgan fingerprints, for example, instead of getting 3 different superclusters and subclusters inside of them, you just get one big sort of blob. And if you do this directly on the label data itself, without actually running it through this set of embeddings, you actually also recapitulate the same structure. So in other words, working with the raw data and working with the representation after it came through the GEN gives a very similar kind of a map. So it is quite interpretable. So what we did next is we asked for 400 novel molecules, how well could this model perform? So now the goal is actually to predict these true labels, right? And ask how well individual humans, we have a panel of 18 people predicting the labels, how well do they predict those labels? And how well did the model predict what labels they would pick? So this next slide is going to unpack all of this. And I'm going to direct you first down to D. D is for a given molecule. Here's this molecule here, it's shown up above. Each individual panelist, say panelist number 6, they gave it a very high rating for sweet. They gave it a somewhat high rating for fruit, a low rating for vanilla, a 0 for powdery, a 0 for floral, and so on, right? And so here the panelists are sorted by how well their correspondence to the mean of the panel. Okay, so the mean of the panel is shown here in C. So here I'm just taking D, I'm just stacking all these together to make C. And so C is the consensus prediction by a human. Okay? And so you're going to ask, how well does the model predict that consensus, right? So and there's the ground truth here. I mean, there is no ground truth, right? Everyone can say whatever they want about what the molecule smells like. So our proxy for ground truth is we take a lot of people, we average them together, right? We say like, this is what the molecule smells like to people in general, respecting that there's a difference between people. And we find is that the prediction of the model, and again, this model has never been trained on this data, it's never seen this data. This model was only trained on the old training data set. We asked it to make a prediction about this molecule, nonetheless predicts a pattern of odor, right? Sweet, fruity, vanilla, powdery, floral, that is better correlated with the mean panelist than alternative types of models, right? So here the model predicts a descriptive vector. There's a correlation of 0.63 with this mean panel rating, and this alternative baseline model is in random forest on physical chemical descriptors, has a correlation of 0.45. What we find is that in general, this model is better than this, right? So we have a model that is now, this was so state of the art as of 2017, and this model is decisively beating this model and predicting on a new task, right? A new data set collected for the entire purpose of running this benchmark test.
Great.
So again, it's hard. We can only do about 400 molecules, but we found, I'm not sure why these slides are out of order, actually. I just showed you that. Okay, here's the summary. So the summary is that if you actually ask how well does the model do compared to the average human panelist. So imagine I run the same test to predict the panel mean, but I actually ask individual raters to do it, right? So I say, okay, P6 was the best. They had the best correlation with the panel mean. P9 was the worst. P19 was the second best. And P23 is the median, right? So this panelist had the median prediction quality relative to the rest of the panel. So imagine it's like a big bullseye, right? In the center of the bullseye is a target. In the center of the bullseye is by definition the average of the panel. And each actual panelist is a dart somewhere around that bullseye. And some of them are really close and some are really far, but the center is by definition the mean of all the darts. Okay. We want to ask whether the model prediction is inside most of those panelists or outside, right? So a good model would do a good job of predicting the mean and do a better job of predicting the mean than the average panelist. I hope that's making sense. So what we found is that the model actually does a better job than the median panelist of predicting the mean across the entire panel, which is to say that imagine you have an odor panel and you have 18 people rating things, and now you have a choice. Do I kick off a random panelist and replace it with a model, or do I not? What this is saying is you'd be better off kicking off that random panelist and replace it with the model, because the model is going to help you converge to that mean faster than if you have a bunch of random panelists. It doesn't say it's better than the best panelist. It doesn't say it's better than an expert perfumer, but it's better than the average person at predicting the mean across this ensemble of people.
Rick?
So in a sense, it passes an odor Turing test. We're defining odor Turing test here. We did not refer to odor Turing test in the preprint or the paper because it's extremely loaded language. But you can imagine defining odor Turing test as, is this model better predicting things than a person? And by that definition, it was. Okay. So you may feel free to disagree with that, but that's just one way of describing it.
Rick?
Yes.
We're running out of time here, so
yep. Then I'll tell you what, I'll skip ahead.
Nice plot.
I'll do global impact. Last 3 slides. Okay. Global impact. Okay. So if you can predict really well what things smell like, and you can predict what things smell like to mosquitoes, for example, can you do something as global impact? So the last thing we did is we asked whether we could take the same approach, use the same kind of fixed model, or actually just made the model more flexible here, and put in a lot more data into the model. So amazingly, the USDA, which is a division of the United States government, during World War II, they were really interested in finding out what kinds of things were repellent to mosquitoes. And they collected a whole lot of data about this, where they had people put their arms into cages, and they put random chemicals in their arms, and they let mosquitoes in. They asked when mosquitoes bit them or not. This was collected in the 1940s and 50s. And amazingly, this data set just kind of sat there, like largely unused for like decades. And this very much more recently has been sort of partly reused. And Google actually digitized all 70,000 data points of this and then brought it into this problem. So they ask is, can you make a prediction based on these kinds of models about what will repel mosquitoes? And there's a high throughput assay here which uses some blood meal and some heated water. And then you can use this data, feed this data back into the model and ask, can you keep trying to generate better and better molecules for repelling mosquitoes from this blood? And what we found was that we could identify molecules, and then we could actually do a real assay with a real person and ask whether in this case, we're a little bit nicer than the USDA was. We don't actually require the mosquitoes land on the person's bare skin, but the mosquitoes can definitely smell the bare skin and go in that direction. And we found was that we could find molecules that are better than DEET and Picaridin, which are in all the, I don't know about Europe, but in the United States, all the mosquito sprays you get have DEET or either DEET or Picaridin in them. We find molecules that are as good or better than those molecules at repelling mosquitoes from a human hand, from a human arm. So that's pretty exciting because mosquitoes that carry diseases kill hundreds of thousands of people a year. So finding things that are even a little bit better than DEET and Picaridin could save many, many lives. So we're able to identify novel molecules to solve global health challenges. Okay, so I want to say thank you to Alex Lichko, who is a leader of the Google team, and to other people on that team who performed the work. Wesley did the metabolic work. Ben developed most of the initial architecture of the model. Jennifer did most of the mosquito work, and Brian did some of the work where we did the prospective validation of 400 molecules. So thank you very much.